Hey CW Bio, Mr. Kennedy here again. This is your Chapter 2 lecture on enzymes. Just want to take a minute to remind you that you can find all of these lectures on my YouTube channel. You can go back and visit them anytime, pause, play as much as you need to in order to get what you need in order to fill out those notes in your notebook. In our last lecture, if you're following the series on Chapter 2, we talked about organic chemistry or biomolecules. So today, we're just going to kind of finish that look at biomolecules by digging a little deeper into the world of enzymes. As we start, I want to revisit what a protein is, and then we'll dig into the whole idea of enzymes from there. So in our last lecture, I basically just copied and pasted some of those slides over here so that you could kind of see or review what a protein was, and we could build on that for how enzymes work. So just as a reminder, proteins are large macromolecules. They're made of carbon, hydrogen, nitrogen, oxygen, and sulfur. Their primary building block, again, is an amino acid. Proteins have lots of functions in our body. You find them in muscle and bone, your teeth, your hair, but probably outside of that, even more important is proteins are what make enzymes, or an enzyme is made of protein, okay? They're also used, just a little side note, for antibodies, which help us fight off infection. Now, let's go through the parts again. Protein's building block is an amino acid. There are 20 different types of amino acids that we use to make proteins, or all the different proteins in our body. And they all start off with the same basic structure, and then the attachment of a slightly different functional group is where we get the variability. Because there's 20 different types of amino acids, there's literally endless combinations of amino acids to give us all the different proteins that we need in order to survive. In our bodies, there's upwards of 5,000 different proteins that we need on a daily basis, just to kind of keep us going, okay? Moving on from there, remember the building blocks of proteins, amino acids, we start to assemble them using something like dehydration synthesis. Removal of water puts two amino acids together. And as we do so, we get a dipeptide. Di means two, right? So we get two amino acids bound together. That's where we get the dipeptide. Normally this isn't a functional protein. Can't really do much with it, but it's a start. Then we get the polypeptides. This is three or more amino acids bound together. These things are huge. All again formed by dehydration synthesis. We just keep linking amino acids together, and we get polypeptides. Poly means three or more or many peptides. Once we have a polypeptide, we got to take our molecule that we've made and fold it into three-dimensional shapes in order to make functional things like enzymes. All right, so there's that. Quick review of protein. Now, we're going to get into the energy of cells and look at how enzymes work. So one of the things that's really important that you need to understand with regard to enzymes is that, everything that goes on in our body, it requires energy, okay? Energy for us that comes from our food is how we get all of those energy requirements met. So when you eat food, your body breaks it down into proteins, carbs, lipids, and all of that. And then that stuff is run through a process called cellular respiration. We'll learn about that later. And that releases the energy from your food so that we can do everything, so that we can move, we can grow, we can replace cells can divide, all that kind of stuff like that all requires energy. Just taking atoms and connecting them to make molecules in your body, that takes energy, okay? So the energy to drive all of this chemistry comes from your food, and we're going to refer to the energy needed to push any of these chemical reactions forward as the energy of activation, okay? It's kind of like the start energy. So the energy of activation is the energy that is needed or required to begin any reaction. This graph over here kind of shows you the overall impact of an enzyme. So most reactions are going to need a little push to get started. Some reactions will actually happen all by themselves. Those are called spontaneous reactions, but those are pretty rare when it comes to the human body. The ones that need a push, right, those are usually the ones that, keep you alive. So the key here is how do we get reactions to go at a pace that's fast enough to keep us alive, right? Well, at the end of the day, there's one easy way to do it, and that is through the addition of energy from an outside source that basically says, here, go. The easiest, simplest form of energy we could add to a chemical reaction comes in the form of heat energy. So if you heat up atoms, they'll move faster. That increases the collision between the atoms and the chances that they will form bonds and react. Only problem is our body's 98.6 degrees. If we heat you up even 5 degrees from 98, we add 5 degrees, you're going to be at like 103. Most people feel pretty lousy when their body temperature reaches 102, let alone 103, 104, right? If we added another 5 degrees and we went from 98 to 108, you'd be dead. Okay, so heating up the human body is not smart when it comes to trying to drive the chemical reactions needed to keep you alive, right? There's got to be a better way. So if we can't heat the body up to add energy to these reactions to make them go, the only other alternative is to make the reactions go using less energy. Okay, that's where enzymes come into play. Enzymes are a molecule that's referred to as a catalyst because basically they make a reaction go without the massive amount of energy that we just talked about, activation energy. So they make the reaction easier to start. The analogy that I like to throw out to kids to think about, most of you can relate to this, is a car. Now, you might not be a driver yourself, but I do know you've ridden in a car. And when you get in the car of today, you've probably seen a parent sit down and literally for some of you, like all they do is push a button, the engine starts and off they go. Well, when I was a kid, my first car, like, hey, we had to fill up the engine with water and then I had to build a fire underneath and wait a few hours for the fire to make the water boil. And then eventually steam got made in the motor and it would start to chug and chug and chug and chug. And I could drive down the road at about 2 miles an hour, right? That took a lot of work, right? As I got older, my car got better. I eventually had like this hand crank on the front. I could go out and like wind the thing up to start it, and it actually had gas in it. Wow, that was cool. Okay. And now today we got push buttons. So the whole idea of making something easier or requiring less effort is in that analogy with those cars. Okay. You could think about first car, all that work I had to do to start the car as a chemical reaction that's not using an enzyme. Okay. Takes lots of work. It's eventually going to start. It's eventually going to go, but I'm going to have to wait hours for that thing to get going. If we're talking about the human body and I need energy now, I can't wait hours for that engine to start. That's where enzymes come into play. Enzymes are like the little computers that you have in the dash of your car today and attached to the motor that make it really easy to start that motor with just the push of a button. So they don't add energy to the reaction itself. They just make it easier to start the reaction by adding something to help the reaction. Okay, so here's a picture of a catalyst working on a substrate. The substrate is the molecule that the enzyme reacts with. Okay, so the catalyst or the enzyme is the green thing, and the reactants or the substrate are the little dots, right? So the enzyme is going to make this reaction go with less effort, with less work involved. That's the key to an enzyme. They don't add energy. Okay, and you'll notice they are also unchanged by the reaction. So I can use this same enzyme over and over and over and over again until like the teeth up here on the enzyme like wear out, kind of like the teeth on a key. Okay, so enzymes themselves, they are proteins, they are catalysts. They're actually a special kind of protein called a tertiary protein, and they bind to reactants in a chemical reaction. Those reactants right here, you can see them. They are called substrates. Okay, so another name for a reactant is substrate. And that substrate binds to an enzyme at the active site, lowering the amount of energy or effort that is needed to make that reaction go. Now, this bond is a temporary bond. The enzyme doesn't stay permanently attached to its substrate. It only hooks up there long enough for the reaction to happen, and then it lets it go. Okay? Now, enzymes lower the activation energy by literally changing the shape of the substrate. They put stress on the bonds of the molecule they've bound to, the substrate, to make it break apart more easily. Okay? By doing so, it takes less effort to get the reaction to go. So in this graph, you can see what happens here. This is a reaction without a catalyst or an enzyme. Eventually, this reaction might build up enough energy to go, but it's going to take a long time to build up all this energy. If I add an enzyme, see what the enzyme did? It reduced the amount of energy that was needed in order to get the reaction to go. Okay, so now the hill I've got to climb is much smaller. Now, next thing, enzymes are very specific in what they will interact with as far as a substrate is concerned. It was once thought that enzymes were so specific that it was kind of like putting a key in a lock. So there was just one key that fit every lock, and that's the end of the story. Well, we've actually revised this idea because if you know anything about locks, right, lock to your house, there's actually more than one key that can open the same lock, okay? You have the key that, kind of everybody has, but then you might also have a master key. Here on campus, I have a key that opens just my classroom. So that's a very specific key. But administrators on this campus have keys that actually open every classroom, including mine. So right there's two keys that look different, very slightly different, and they open the same lock. Okay, so we've revised this hypothesis to what's now called an induced fit hypothesis. So basically the idea that we think is going on at the enzyme substrate level is kind of like this. Here's the enzyme, this pink stripy thing with the divots in here, which are the active site. And you can see the substrate has kind of a matching set of appendages here that will fit into the divots on the enzyme. But they don't look like they're going to fit perfectly because this end is round and this indentation here is flat. That being the case, look what happens, okay? When the enzyme is shoved into the substrate, we get what's called an induced fit or a stressed fit. Basically, these flat ends are going to put pressure on the tips of the substrate, weakening those bonds and causing the molecule to break apart with less effort. Okay. This also means that this enzyme could actually be used on more than one substrate as long as the substrate actually has kind of the same general structure there. Okay. We could keep using it again and again and again. Next thing, enzymes are rapid. They work fast. Okay. They'll bond to the substrate, catalyze the reaction, move on to the next reaction again really, really quick. Like literally thousands of reactions per second. It's insanely fast. That's pretty cool. That means that I don't need tons and tons of enzymes floating around in my body just to kind of keep me alive. I can have a very small amount and still perform all the reactions that I need for life because the small quantity of enzymes do their job really, really fast. So that's what this slide's all about. Enzymes are required in small quantities. Only takes a very tiny amount of an enzyme to speed up a reaction as little as 1 part per million. All right, moving on. Next, enzymes show what's called competitive inhibition. You got these enzymes running around in your body, and it would be really great if, we could control what they were doing. That's where competitive inhibition comes into place. Okay, it's a means of regulating the way enzymes work. So if you look close to this enzyme, you're going to notice there's a substrate up here, a reactant, that's fitting into the active site of this substrate, and a reaction is occurring. Okay, right here, see this little blue dot? Okay, if this little blue dot gets to the active site before the funny little green-shaped jelly bean thing gets there, that's called competitive inhibition. It's kind of like whoever gets there first wins. A great example of this is the reaction in our red blood cells between hemoglobin, the molecule that carries oxygen in our body, and oxygen or carbon monoxide. So if we have oxygen in our bloodstream, hemoglobin will try to bond to it. If we have carbon monoxide in our bloodstream, carbon monoxide can also bond to hemoglobin. And so as the rule was laid out for you, Whoever gets there first wins. If carbon monoxide gets there first, it's going to bond to hemoglobin and block oxygen from bonding, which is bad for us because we need oxygen. We can't do anything with carbon monoxide, okay? So good idea to avoid that. So the definition of competitive inhibition is two or more substrates competing, let me get that out of the way there so you can see it, for the same active site on an enzyme, okay? And the example, carbon monoxide on hemoglobin. Carbon monoxide will fight for the same active site that oxygen normally binds to on a hemoglobin molecule. Cool. Now, requirements for enzyme actions. Remember, enzymes are proteins, and normally, like, anything that's true of a protein, true of an enzyme, because, enzymes are proteins. So anything that messes up a protein or affects the, quality of life of a protein, it's going to affect the quality of life for an enzyme. Enzymes will only work within specific temperature ranges, pH ranges, and all of that. I guess they're kind of like us. We all have like certain temperature ranges that we like our room to be. If our room's too hot, we're not happy. If our room's too cold, we're not happy, right? So enzymes are the same way. So what I've got for you down here is a series of graphs just to kind of like show you what I mean in terms of what enzymes like with regard to the different conditions in which they work. And you need to pay close attention to these graphs. You need to like memorize these graphs, tattoo them to your brain because you will see them on your quizzes and tests. Okay, so right here we've got the temperature ranges that enzymes prefer, specifically those in the human body. And check this out. As it gets warmer, enzymes work faster, but there's like this limit. And if we go past that limit, look what happens. The line goes whoop down to the bottom, and all reactions stop. So that basically means, in terms of the human body, I can't just heat you up and expect, all your enzymes to work. If your body gets over 108 degrees Fahrenheit, which is around 42 degrees Celsius, your enzymes will literally stop working, okay? And if you don't have enzymes working in your body, then the reactions shut down and the rest is history, okay? pH is the same way. Check this out. There is a range, a pH range that your enzymes like to work in. And if we go too far to the acidic or too far to the basic range, again, your enzymes will shut down. This last graph is all about enzymes being saturated. There is a pace at which enzymes can't work any faster, and that's what we're seeing down here. As I give enzymes work to do, they're going to work faster and faster and faster, but at this point, they're like, hey, I don't care how much you give me, I can't go any faster. Okay, it's kind of like me giving you homework. If I gave you one homework problem to do and I told you it was due in an hour, some of you would do it right away and then you'd sit around for 59 minutes. Okay, others of you would wait 59 minutes and then right before it's due, you'd do it. At any rate, you're not going to be super stressed about doing one homework problem an hour. Well, let's flip the script. What if I told you you had 50 problems to do in an hour? Okay, now you're doing about one problem a minute. So now you got to work faster. So this is where you would be on that curve. You'd be working faster to try to keep up. What if I said you have 1,000 problems to do in an hour? You'd probably look at me and go, Mr. Kennedy, are you nuts? Like, I can't get all that done in an hour. I'll try, but not going to happen. So you're going to work as fast as you possibly can and basically just stay at that level, and you're going to get done what you can get done. Enzymes do that too. We call that the saturation point, okay? All right, so again, enzymes are proteins. They're bound by the restrictions that influence all their biomolecules, temperature, pH, all that good stuff. If we violate the temperature range they prefer or the pH range they prefer, they will denature. The word denature means destroy, okay? Now, usually denature is something that happens only on the top ends of these violations. So if it gets super hot, that's where the enzyme falls apart and dies. If it gets super cold, it doesn't necessarily denature. It's just going to work really, really slow because it's like freezing to death, okay? So if you warm it up, like that cold enzyme could still work. If you had something that was super hot, it's destroyed. So you can't cool it off and be like, oh, it's okay now. Nope. It's ruined. It's destroyed. It's gone. You broke it. Okay, so there's no going back and fixing it. The only thing you can do is make a new one. All right, next thing. Enzymes need cofactors and coenzymes. Cofactors and coenzymes, these are like helpers that help your enzymes go. They come in two forms. Some are organic, some are inorganic. If it's organic, we're talking about things like vitamins and, um, big complex organic molecules. Those are our coenzymes. If it's a mineral, maybe it's iron or something like that, that's a cofactor. Okay, so your coenzymes are organic and your cofactors are inorganic. All right, last piece down here is just a little bit about how we shut these enzymes off, the regulation of enzymes or keep them in check. And that involves a thing called an allosteric site. So enzymes have active sites. They also have allosteric sites. Looks like the active site, but instead of like catalyzing and getting a reaction to go, this just like shuts the enzyme off. So allosteric sites are all about inhibiting factors, right? They will actually stop an enzyme from working. And this is kind of cool because you might need to actually turn an enzyme off. Enzymes are not smart, so they don't know like the whole concept of like go versus stop when I'm needed. They just, if you put work in front of them, they just go, okay? So you need to be able to turn them off. Now, one of the things that we're going to spend some more time talking about later is this idea of feedback inhibition. And that's where this allosteric site and its regulation of enzymes really like shines. So as enzymes produce products, the products of the reaction feed back and they bind to the allosteric site and shut the enzyme off. More to come on that on another day. All right, gang, so that's your lecture on enzymes from Chapter 2 for CW Bio. All right, catch you next time.